Hey microbiologists! I'm so excited for our class today because we are talking about microscopes. We'll get to spend a lot of time in lab playing with them and actually doing stuff with them, but we're going to talk a little bit about how they actually work, a little bit of understanding some of the theory and functions of a microscope. So let's start off with the metric system. Nice and important to use. All that stuff you're supposed to say. There you go. You can review that. When we're looking at microbes, something like a tick is something you could see in real life, right? If you had a tick on you, you could see it. As we get smaller to things like eukaryotic cells, red blood cells are one of the smallest cells in our body. They're too small to see by themselves, but a microscope could pick it up. E. coli bacteria, way too small to see. They're about a micrometer long. These are things that light microscopes can see. So this here is where light microscopes can see things. Then down here to viruses that are around 100 nanometers. An electron microscope could see that. So we won't be able to see this with our microscopes, but an electron microscope could see that. And even on down to things that are around a nanometer, like a double helix of DNA. So this is the scale we're going to be using our microscopes for. We'll actually do a little practice with a tick, so we'll get a little bit towards there, but this is the scale we're looking at, mainly where our light microscopes are going to stay. If you remember back when we were talking about Leeuwenhoek in the 17th century, when he was using a microscope, he was using what's called a simple microscope, which had only one lens. So really, it was kind of a jazzed up version of a magnifying glass. You could get magnification up to about 300 times, which is actually kind of impressive. That's not bad at all. But it had this little bubble here that was the lens that you would look through that would magnify. This little pin that you could put a little sample on it. So you would do things like look at a fleck of pepper, look at some of his semen, look at teeth scrapings, whatever. This little screw, you could focus it closer or further away to focus it, and this would move it up and down to help position that stage at a place where you would be able to look at your object. We use light microscopy in lab. Light microscopy uses visible light to observe specimens. So it's going to take light that exists as a wavelength and use that as a basis for magnifying our images. There's lots of different types of light microscopy, but what we're really going to talk about is compound light microscopy, dark field microscopy, bright field microscopy, and fluorescence microscopy. Those are really the big ones here. In compound light microscopes, you have a series of lenses, ocular lenses and objective lenses. Ocular lenses are the part you look through. They're mostly around 10x. And objective lenses are the part that you can change to have increasing levels of magnification. In our lab, they're going to be 4x, 10x, 40x, and 100x. And to find out how much you're magnifying between both lenses, you multiply together your two lenses. So if you're using a 10x ocular lens and a 4x objective lens, you say 10x times 4x, your total magnification is 40x. If you're using a 10x ocular lens and a 100x objective lens, you say 10 times 100, your total magnification will be 1,000 times. In compound light microscopes, we use visible light to illuminate our samples and allows us to look at small things and see some of their fine details. Here's an example of a microscope we use in lab, and here's some of the different pieces. Here, this part you look through is called the ocular lens or the eyepiece. We will stick with calling it the ocular lens, and this images the image. So whatever comes up to it, it is going to magnify it again. This is the body tube. We won't talk about that much. The arm, that's what you carry it with. These are your objective lenses. So they go from smaller to bigger, gradually magnifying in greater and greater amounts. Their main job is just to magnify your image. This is the stage, which holds your slides. You can see the slide sitting on here. And it's got these little clamps here on either side that hold it in place. So you just put this slide right up against this wall, and this clamp just moves out and forward to just position and hold it in. The condenser here and the diaphragm, these really focus the light to go through your specimen. This is the illuminator, which is your light source. In our microscopes, they all have a light that you can turn on and off, you plug in. Although some microscopes will have just a little mirror here that you have to reflect the room light onto it. These are your focusing knobs, a coarse focusing knob and a fine focusing knob. So the coarse focusing knob is what you'll use initially with your 4X objective lens. It moves the stage very quickly. So it's just to initially get your slide in focus. After you've focused it on the 4X, the only knob you use is your fine focusing knob. And that gives you a little bit more careful control of how quickly the stage is moving up and down so that you don't accidentally just pass right over your sample. So once you kind of see something with your coarse focusing knob on your 4X, then you focus it with your fine focusing knob with your 4X objective. And then as you increase your objectives, you're only going to use a fine focusing knob. Then the base here, this is the part where the base of the whole thing, where it's sitting. Here's a little light control. On the other side, there's probably a light switch to tell you to turn the light on and off. Okay, so in a microscope, one of the important things it does is magnify your image. It makes it bigger. But if you've ever had like a camera that had really poor resolution, maybe an old cell phone, or you've just tried to copy and paste a little image off the internet that was just a small little picture, and as you try to blow it up, you don't have a clear image. You just have a bunch of pixels, and those individual pixels get bigger and bigger and bigger. That's because your image doesn't have good resolution. To have a nice clear image, you need good resolution. Resolution is how well your microscope is able to take two different points and tell that they are two separate points. So 2 points that are far away, it's pretty easy for a microscope to tell that they're two separate points. But as they move closer and closer, eventually they're going to start to just blur into just being one blurry spot. So different types of microscopes have different types of resolution because they're able to tell the difference between 2 points to say those are definitely two different points. The resolving power is how well your lenses can tell the two different points are absolutely two different points. So if a microscope has a resolving power of 0.4 nanometers, that means that 2 points have to be at least 0.4 nanometers to tell that it's two separate points. When they're closer than that, they're going to blur together. The way that your microscope has resolution is based on what kind of wavelength of light it's having. So light is traveling in a wave, and as wavelength gets bigger, your resolving power gets bigger. So smaller wavelengths of light have a smaller resolving power, so they have much greater resolution. So as we are using light with smaller and smaller wavelengths, you're going to have more and more clear images at higher power. You'll have greater resolution. Another thing that's important when you're looking at a microscope is being able to have your cell not just blend in with the background. A lot of cells are almost colorless, just invisible almost. So we're going to have to stain them so they look different from their medium, from their background. We don't want them to blend right in with the glass slide or the fluid of the cell or the fluid of its surroundings. We want it to be able to tell very sharply, this is my cell here. And another image to take into consideration when we talk about microscopes is refraction. So if you look at this glass, you have this straw. That straw is actually one continuous straw, but it looks like it's bent right here at this interface between the water and the air. So as light moves through different substances, it moves differently to cause this refraction, to cause this to look like a bend. If you're looking at a cell and you have that effect, you're going to have no idea how the cell actually looks. You're not going to know, am I actually looking at what the cell looks? Does this cell actually have a little bend here? Or is this just refraction? So refraction is something that we want to minimize. Refractive index is how well different mediums bend the light. So water and glass will bend the light differently than air will. It could bend the light so much that you entirely miss a cell even. So To minimize refraction, we use what's called immersion oil. And actually, a lot of times in our lab, we just use pure water for it. That water has the same refractive index as glass, so the light will go through the lights of the glass, through the cover slide, through this immersion oil or water, right into your microscope in one nice solid straight line, instead of the light going through the glass, hitting the air and bending away. So this is what we don't want. This is what we do. We use that water to minimize the refraction of the light so that we get a nice, clear, focused image without this kind of bending going on. Okay, so we've talked about what lenses a light would pass through in a compound microscope. What does it mean when a microscope has a resolution of 0.2 nanometers? Be able to identify and describe the various parts of a microscope. So if I have a picture of a microscope, or if I have a microscope set out, you could tell me what each part was. Or if I asked you, what does this particular part do, you could tell me. How do you calculate magnification? And then also let's talk about refraction. What does refraction have to do with any of this? How do we minimize refraction? Okay, I will see you back in our next video where we are going to talk about different kinds of microscopes. See you soon. Bye.